help to produce hydrogen production. Now, if I want to draw your attention about T. maritima and fermentation. So as you see in this table, there are researchers that they have done research on studies on using T. maritima, but they use pure substrate for production of hydrogen. Of course, they are successful in it, but till now they are using the pure substrates such as glycerol, glucose, xylose, cellulose, and recently, maybe in 2018, they started to use on pure bio waste. They have used on fruit and vegetable waste, but still they have not challenged the lignocellulosic bio waste. Why is this important to use the lignocellulosic bio waste? Because the plant cell wall is mainly consists of cellulose, lignin, and hemicellulose. And this structure, the binding of this structure makes it very difficult to degrade, because lignin is resistant to biodegradation because of this cross-link network structure that it has. And penetrating through this lignin structure is very difficult. Of course, many pretreatment processes have been done, mechanical, chemical, biological pretreatments, but mostly these pretreatment processes cost maybe 40% of the total process. And beside that, if they want to use any alkaline or acidic pretreatments, again, they are posing addition of extra hazardous material waste in the environment. Therefore, always the challenge is to choose a green technology. For this reason, the bacteria comes into the picture. So if I want to combine all this information together, the experiment that we have done at our Biofuel Institute, we have chosen corn stover as a biomass material, lignocellulosic waste, and we have used Thermophilic maritima, which can grow up to 80 degrees. The special optimal basal medium also have been designed in our school by my professor, Professor Shah, and extracted this specific hyperthermophilic enzyme. We designed this bioreactor using our biomass, the bacteria, introducing the enzyme. Of course, this will be an anaerobic process to degrade the biowaste and towards biohydrogen production. Through this process, we have obtained very good results, actually. The hydrogen, we could obtain the hydrogen up to its optimal level. Near to the optimal level, of course, it wasn't full. The hydrogen was measured through the syringe method, and it was checked with gas chromatography. And as you can see in the picture, before addition of any bacteria or enzymes, we have put these reactors in incubator for 12 or 24 hours. And as you can see through the process from left to right, the color of the biomass is changing. It means it from this light brown, it is changing to dark brown. This means some process is happening, some degradation is happening, but there are some stickiness stuff is also visible, which we have to check for further studies. While we have extracted the enzyme from the cell wall of T. maritima, also we got a very good result, because we saw that the enzyme activities, xylanase especially, was very high in the cell wall process, and high protein concentration of xylanase and pectinase, which makes us believe that this definitely can be an approach to degrade the lignin structure of our corn stover. Also, when we have checked the enzyme activity, we have checked different enzyme activity, cellulase, xylanase, and pectinase, and we had industrial alpha xylose activity. But to our surprise, within 1 hour of experiment, everything was well. Most of them showing increase, but after 1 hour, they are decreasing pattern. That's why this result and the results of previous slide, they are correlating with each other, that there is some inhibiting factor in our process, which makes us decrease in its activity. Either it is excess of hydrogen, which of course we try to maintain it, or lack of the food, or releasing some other substances which is unknown for us. Therefore, we continue the study to check what other monomers are releasing during the the incubation time. And we have used HPLC method. Of course, because the monomers are low absorbance usually, and it cannot be detected in column C18. That's why we have used derivatization method. In this method, there is a label will be used, amino benzoic acid. This amino benzoic acid will make as a sheaf base, will attach to the to our sugar polymer and makes the combination will be a secondary amine that can be detected in our HPLC result. And as you can see in this figure, the thick and the tall peak at time 35 is actually our label. This is the aminobenzoic acid label, ABA, which means that the process and the amination has been done successfully. Therefore, the peak beside to that, around time 30, it is our standard glucose standard. Therefore, we have run other standards and we come up with this wonderful result. Apart from the glucose, which is of course in the process, it means that the structure is degrading, producing glucose. There are two other polysaccharides monomers we can detect, which is galactonic acid and galactose. Therefore, this can be an assumption that during this incubation process, apart from glucose, which is releasing, galactonic acid or galactose also is releasing in a good amount, which may making this inhibition factor for our process to decrease the process and decrease the hydrogen production. This results is till now, and it is still continuing. And we are trying to still battle this process and come up with the best results. So far, this is a very unique and very unique results we have obtained. So I welcome other researchers or scholars if they want to join our team, if they're interested to have further studies in this topic at Jiangsu University School of Biofuels. Very welcome. So to conclude and compile today's talk, we have told, or we have said that this degradation of corn stover using anaerobic hyperthermophilic bacteria, Thermophoca maritima, is possible without doing any pre-treatment process, we could see. And also the process can be enhanced while we have extracted its relevant enzymes. And more and most important one is to detect the inhibition factors such as galactonic acid. So it may be the reason for this stickiness stuff and decreasing our process. And of course, to apply its relevant enzyme, extract the relevant enzyme for further studies. As the future challenges, always we have future challenges in our environmental studies. So selecting and introducing this novel biomass degradation process via microbial consortia and enzymes, of course, to detect that which other pathways we can go, like we can inspire from nature having lignocellulose feeding animals we have in the nature. So we can detect the enzymes in the gut microbiota, detecting microbial consortia, effective enzymes, and towards, of course, having a green energy biofuel. So as environmentalists, we always think
يعني الآن
waste and well. Thank you
طيب
very
حق الصباح
much. Thank you very much for your time and listening to my talk. And I would like just to close this talk with a short sentence saying that always be like eagle, be unique, and love to face challenges in your life. And most importantly, always
brother
thank you.
170 now 30.
Thank you so much, Dr. Barfin Fatima. Thank you so much for your clear representation. Now
Thank
we
you.
will open the season for answer questions. If there is any question, you can raise your hand, and Dr. Naif, you can select and unmute if you have any questions, please. What is our friend Dr. Ali Shahiri? His chemistry. Dr. Ali, Ali Shahiri. Is there any question? Okay. Doctor, about the bacteria, do you determine the temperature? What we said, thermophilic means it is like bacteria temperature, high temperature. Do you know what's the degradation, the temperature, which is degraded the cell wall?
Yeah, that's the research that we wanted to check. So now, of course, when you put it in a very high temperature, they will degrade. But this process is to put it in 80 degrees Celsius or 85, that they can maintain the temperature of thermophilic bacteria also.
So you optimize the temperature or not yet? The optimization of temperature.
Already it is optimized to growth for the bacteria. So the optimized temperature for this bacteria is 80.
Okay. What is about the difference between biofuels and the normal fuel from the petrol? Is this the same activity or how?
Different between biofuels and
Yes, and normal fuels like the what we get from the station, the quality.
Of course, biofuels, of course, yeah, of course, biofuels is based from biomass.
Okay.
So different generations of biofuels we have also, depending on the waste that you're using.
For the lignocellulose enzymes, we can get it from any other fungal, or just bacteria.
Yes, of course. From fungal also you can detect. There are many fungus that they are having this degradation ability. You can see it in jungle, woods. Specific fungi are working there to degrade the wood. So of course they can be isolated from wood also.
Thank you, Doctor. What is about our friend Prof. Abdurrahman Humaid? Dr. Abdurrahman, you're around?
Dr. Mohana, there's a question in the chat box.
Yeah, maybe.
One of the participants would like to know, apart from the corn stover, what other biomass that have you tried? Or do you suggest we can try, for example, bagasse, rice husk, or would conditions change? This is from Asha. Yeah?
Yes, of course, that is a good question. Of course, any lignocellulosic biomass can be checked. So far we have only checked on corn stover. And still, if this can be successful, of course, you can check with other lignocellulose biomass. As much as hard this lignin process is, you can penetrate the lignin, then you will say bravo. We can do for everyone, each ways.
There is also one question. They said, how can we obtain the bioreactor used your experiment?
Say again, sorry. How can I?
Okay, how can we obtain the bioreactor used in your experiment?
The question is not clear to me, but the bioreactor which we use is like the anaerobic bioreactor, as I show in the picture, 100 ml working reactor. Because this is the lab scale experiment.
There is another question from Brother Nasser. He said, what is the yield that you may obtain from this process?
Yeah, the optimized yield is like, of course, we will wish to have at the optimal or near to the theoretical level, which may be very difficult. But as other researchers have been done up to 280 maybe mole per liter of glucose, perhaps we are also wishing to reach to that optimized good amount of hydrogen.
Okay. Thank you, Doctor. There is also a link for the certificate of today. Please don't forget to fill the form and you get your certificate for attendance for today. Thank you, Dr. Fatima, for your very nice presentation.
Thank
If there is
you.
any
Thank
question,
you, everyone.
Yeah, Dr. Muhammad, there is some of a question also in the chat.
okay, you can try and read because I don't see clear here.
Could you please, okay, there is one question here. Can we use such biotechniques in biotreatment of plastic residues?
That is really good question. I wish.
Yeah, he mean, can we
Yes.
use the plastic as biomass or substrate
course.
for biofuel?
Of course, we can use plastic, and recently, like the focus is, it is a really good question because now plastic is in the main focus also. But to degrade this plastic, there are researches have been done also, some bacteria they are using, but if the question is to then lead into the biofuel or biohydrogen production, it can be a good new approach.
Okay, there is one question. Thank you for your answers. For actually, so many questions here I can saw. Are you planning to try pilot scale for your experiment maybe?
Yes, inshallah, if we can make it successful to up to the the lab scale, of course, this will be a very good opportunity to scale up as industrial scale.
Yeah, there is one asked about how we can isolate the retina. Maybe this one is bacteria or what? I don't think so.
The same bacteria which is used.
It's okay. So isolation of bacteria is depend on the source. Okay, can we use such bioactive? This one I already read it. Thank you. I have a question. Can we use such biotechniques in biotreatment on plastic? Already read. Okay, there one question from Esther here. If there is any side effect to human health after the use biohydrogen production, any health effect for the human?
It shouldn't be. That's why it is bio. That's why we are leading into the green technology.
Yeah.
So we want to replace it with the normal fuel because it's increasing GHG in the environment. So the main concept of this research is to reduce this effect.
Okay, Dr. Muhanna, come back to you. If you have someone want to ask.
There is no question. Thank you so much, Doctor. Thank you, all of you. Inshallah, for next season, it will be November 6th, Thursday, with Associated Prof. Dr. Iqbal Zakan from Norway. It will be about natural antioxidant, natural defense against oxidative stress. Dr. Maya, there is any comments?
This is a very good sharing from Dr. Praveen. I'm very glad to have you here to join us today. I hope that we continue collaboration with Dr. Adel and Dr. Muhanna and everyone. Thank you so much. It's very nice.
Thank you, Professor. Inshallah.
Okay, thank you, everyone. Going back to Dr. Muhanna.
Thank you so much. Thank you all. Just don't forget about link to fill, and you get certificate. Saddam, Dr. Saddam Hussein, do you have any question?
Where is he?
No, thank you. I was actually in the training show. Just I come in the middle of this session, so I couldn't get the whole idea. Maybe next time, inshallah. Thank you so much, Dr. Muhanna.
Saddam Hussein, you can give us like, you know, he's a veterinary, surgery veterinary. You can give us like zoonotic microorganism and next time, inshallah.
Inshallah. Maybe, inshallah, if we have time, no problem. Thank you so much, Dr. Muhanna.
Prof. Abdurrahman is around. Abdurrahman Humaid. Dr. Khairullah Al-Ghazali.
Yeah, he's around.
Dr. Khairullah is our speaker. I don't know, it's not mute now. You can say no mute. You can
yes,
open.
Dr. Muhanna.
Yes, Assalamualaikum, Khairullah.
Alaikum salam.
So, you have any comments, any question?
Thank you. Everything is clear. Thanks so much for the Dr. Fatima. Very nice presentation and very nice talk.
Okay, thank you, Doctor. Thank you.
Thank you, يا دكتور. Thank you. فيها.
Okay, thank you for all. We can now close.
Yeah, please, okay, Doctor, before closing, please don't forget to register using the link, please, all.
Thank you, Dr. Naif. Thank you so much.
Thank you. Well done.
Yes,
Okay,
Dr.
so maybe
you will drop the link in the group of WhatsApp or what?
The chat.
No,
the chat.
the link here in the chatting.
In this chat, okay, just
chatting, please. Okay, I will share it.
Wait. Okay. You can share it in the WhatsApp. Okay, I find it. Thank you.
Okay. Thank you, everyone.
Thank you, Dr. Adil. Thank you so much.
Welcome.
Inshallah, our next season in the 6th of November with Dr. Iqbal.
Yeah, Dr. Barbin, she will be our moderator for that
yes,
sharing
Dr.
session.
Barbin, inshallah, our moderators.
Inshallah. Thank you, Dr. Muhanna. Thank you, Dr. Adil.
I will send you the CV and you can introduce here.
Okay, sure.